Okay, so here is a brief overview of Chapter 6.1 on digestion. So what we need to cover, we're going to start with talking about mechanical and chemical digestion, taking in food and breaking them down into components which then our body can use. For our body to use them, they need to be absorbed into the bloodstream, meaning passing out of the digestive tract and into the blood. And finally, assimilation, where those particles join in to become part of our bodies. So taking a step back, looking at mechanical and chemical digestion starts with taking a look at the digestive tract. At this point, you should pause and make sure you're able to label everything that is there. Okay, so we're going to walk through organ by organ as if you were a piece of food. So if food were to come in, it's going to come in first through the mouth, go down the esophagus, wait in the stomach, continue to the duodenum, throughout the small intestine, into the large intestine, and finally out the rectum and anus. And now we'll take a look at the function of each part, starting with the mouth. The mouth performs mechanical breakdown in chewing, as well as chemical breakdown because of the salivary amylase present in the saliva, as well as many other enzymes that help with breaking down foods. Food then goes into the esophagus. The esophagus uses peristalsis to move food towards the stomach. And we get to the stomach. The stomach acts as a temporary storage for food, as well as does a lot of churning for mechanical digestion. The biggest part of why the stomach is so important is because of the digestive juice which the stomach glands secrete. And one of those components is hydrochloric acid, which is going to make it a very acidic environment, about a pH of 1 to 2, meaning that you're killing off bacteria, you're denaturing the proteins that do enter so that they're even easier to digest, and pepsin becomes active at that really, really low pH. So we're going to talk about pepsin here in a couple of minutes. From the stomach, we go to the first part of the small intestine, called the duodenum. In the duodenum, we have secretions that come from the gallbladder and the pancreas, and again, we'll get back to that here in a minute, but those are going to be dumped into the duodenum first, and then we move to the rest of the small intestine. So the small intestine is really where most of your absorption is happening of nutrients. And because of that, you have a ginormous surface area. There's a lot of folding. We're going to talk about that here in a minute as well. We should be thinking about things like villi and microvilli. And there are going to be three parts: the duodenum, jejunum, and ileum. The duodenum we just talked about. The jejunum you're not really going to need to know for the IB exam. And you'll need to recognize the word ileum, but in terms of actually citing those three, you really won't need to see that. But the ileum is one of the objective statements for you, asks to be able to draw a cross-section of the ileum. All that really means is draw a cross-section of the small intestine, and I'll show you how to do that. Then from the small intestine, we go over to the large intestine. Zoom out here so we can see that. Let me see. Okay, food has gone from the stomach here in throughout the small intestine and finally into the large intestine right here. So from the small to the large, and remember that appendix is hanging down off the end of the intestine here. So this whole piece is the large intestine. Most of the large intestine's job is reabsorbing water. up until this point, we've had a very soupy, liquidy consistency, and so being able to absorb most of that water is going to allow you to have a semi-solid feces, which then is stored in the rectum before being excreted through the anus. So there you go. Everything that you're looking at right now is part of the alimentary canal, the parts where food is actually going through. So now we need to take a look at a couple of accessory organs, or organs that are responsible for making something that aids in digestion, but isn't necessarily where food is going. We can start right at the beginning with 2 salivary glands, which enter into the mouth. The large orange structure right here is your liver. The green one okay, so this is your gallbladder, again on the underside of your liver. And this here in purple is your pancreas. And all of those are, the gallbladder here and the pancreas are both leading into the duodenum. So all of their products are going to be leaked out into the duodenum. So let's take a look at exactly what enzymes each of these are producing. Going up to the salivary glands. Here we're looking at that salivary amylase. You saw we already talked about that a little bit with the mouth. The liver is actually going to be responsible for making the bile, but that is going to be stored in the gallbladder. And then we move over to the pancreas here. It's a long kind of feathery organ that is right underneath the duodenum, and then as the small intestine curves, you can sometimes kind of see it, but typically it breaks up in a cadaver, so very difficult to see. And so that pancreas is going to be secreting a couple of enzymes which are really important. First of all, insulin is actually a hormone. We'll come back to that later, but when you're thinking pancreas, then your insulin should definitely be the first thing on your mind. In addition to the hormones, the insulin, there we go, that is produced by the pancreas. The pancreas is also making amylase and trypsin. So in terms of digestion in this chapter, those are going to be the really important ones that you'll need to know. So again, let's take a look at everything here, piece by piece. And so we've got the mouth, the esophagus, food's coming down, ending up in the stomach. While in the stomach, you're going to have the digestive juices excreted on them. That's going to be hydrochloric acid and pepsin, which is going to start the digestive system and actually really get it going. Food's going to move into the duodenum, where it's going to have juices that have been made by the liver, secreted by the gallbladder and the pancreas. Let me clarify that. The liver is making the bile, the gallbladder is secreting it, and then the pancreas, separate from that, is making amylase and trypsin. So in the duodenum, your food is now going to be covered in those materials as well, to really further break down the foods that you're eating. You're going to travel through the rest of the small intestine, where absorption is going to happen, where we're getting all of those nutrients into the blood system. Then we move into the large intestine. Most of the nutrients at this point that you're going to digest and absorb have already been absorbed. So really at this point, you're just getting the rest of the water out so that you're not stuck with, diarrhea, because that sucks. Finally moving out of the rectum, out of the anus, where you can finally excrete whatever is left. And we'll talk about that next year in higher level. So now we have to spend some time talking about what each individual enzyme is doing. Okay, objective 6.1.3 asks you to be able to state the source, substrate, product, optimal pH for any one amylase, protease, and lipase. So a couple of things here that we want to make sure that you keep in mind. Anytime you see that ending ACE right here, that ACE, you know that you're talking about some type of enzyme. So we're going to be taking something and allowing it to happen at biological temperatures without having to add in a huge amount of activation energy, right? So if we take a look, let's move over to the side here. And remember our reaction curve here, that you're going to start with a certain amount of energy in any given chemical compound, and for the reaction to happen, you have to add some amount of energy. That's called the activation energy, and then the molecules change, and then you've released energy, right? So you're at a lower energy state. Well, the problem is that this activation energy, E sub A, activation energy, this activation energy is typically too much energy for what our bodies can handle, meaning that you'd basically light yourself on fire before being able to reach that amount. So all an enzyme is doing is lowering the activation energy. So it takes less energy to allow the enzyme, or to allow the reaction to go to completion. And the way it does this is you've got some kind of enzyme, and that is going to fit some kind of substrate. Remember, your substrate is just another name for your reactant. So here's your substrate. And the enzyme is going to be changing the shape of the substrate and kind of clicking it into position to allow that reaction to happen without having to add that huge, ridiculous amount of activation enzyme. So we're going to reduce how much energy that we need to make that chemical reaction happen. Okay, so again, we're looking at one amylase, protease, and lipase. And the names of each of these is also going to give you a hint. An amylase might be the hardest one to remember. That just means we're taking some kind of sugar and breaking it down. But a protease, that makes sense. Protein, protease, right? So protein, meaning a protease is going to break down a protein. And like you could guess, a lipase is going to break down a lipid. Okay, so let's take a look down here. And again, we're needing to cite the enzyme type, amylase, protease, and lipase, with an example, the source and the substrate, meaning what is your substrate, what is the actual chemical that is getting changed, the source, meaning where is it coming from, and an optimum pH. So one more graph that we're going to do over here, because remember, for every enzyme, this would be your enzyme activity, right? Or the rate of the reaction. And for every either pH that you have or temperature, you've got an optimum, right? And that is the fastest that it's going to go. And that if you reduce from the optimum, it goes slower. And then if you increase from the optimum, it goes slower. In thinking about temperature and pH, when we go too high, you can actually have denaturation. But even before that happens, you've got this zone where the speed decreases, or where the rate decreases. So decrease, but not because of denaturation. Once denaturation happens, then we go back to talking about proteins, because enzymes are proteins. And we've got those 4 degrees of protein structure. Primary, so our primary, secondary, tertiary, quaternary. And so that primary is the sequence of amino acid. Those secondary and tertiary structures, that's all the folding. And so when you go too hot, or your pH is too out of balance, either too high, basic, too low, acidic, when that gets too off from where the protein wants to be working, then that secondary and tertiary structure is going to start to break down or denature. Okay, so back over here, we've got our enzyme types, examples, source, substrate, product. Substrate meaning what are you starting with? Sorry, let me try that again. What you're starting with, what you're ending with, and the pH that is going to allow this all to happen. So here we go. Remember these ones. For an amylase, this is very easy. The name's right in it. So we can think of our salivary amylase. Amylase. And as the name would suggest, the source is going to be the salivary gland. Okay, so for this, we're starting with starch, and we're breaking it down into maltose. Okay, now think about your mouth for a second. What is the pH of your mouth? It's not very, very acidic like your stomach is, but it's neutral. And for that reason, our salivary amylase is going to work best at a neutral pH. Okay, all of that makes pretty good sense, I hope. Remember, another amylase would be pancreatic amylase, which comes from the pancreas, but your optimal pH does change a little bit. Okay, moving on to protease. We can look at pepsin. And remember that the source of pepsin is your gastric gland. Another name for gastric gland is your stomach gland. So the gland of your stomach. And so for our gastric glands making pepsin, we're going to be trying to break down a protein. And we're going to, instead of breaking it all the way down into amino acids, we're just breaking it into short polypeptides. Okay, meaning that maybe 5 to 10 amino acids long, so fairly short. Now, if we're thinking about the gastric gland, i.e. the stomach, we want to be thinking, what is the pH of the stomach? And like we said before, that high amount of hydrochloric acid, HCl, very, very acidic, which means that pepsin is working best at a pH of around 1 to 2. So if you put pepsin in your mouth, it's not going to break down anything. It needs to be in a very, very low pH environment. And finally, we're going to look at pancreatic lipase, is the particular lipase that you should be familiar with. And if you can't figure out where this comes from, then ay caramba. Okay, comes from the pancreas. And like all lipases, we're breaking down lipid, fats, and this is going to break them down into their monomers, which are glycerol and fatty acids. Remember that back from our chemistry chapter. And now this is happening in the duodenum, because as we remember, the pancreas is excreting its products into the duodenum. And the duodenum does not have that really, really low pH, that very acidic environment. The duodenum actually goes to a slightly basic condition, meaning, that's really ugly, meaning that we're thinking of a pH of roughly 8, slightly more basic than neutral. So spend a little time looking at this. Make sure you know this chart. Make sure you can recreate this. If I were you, right now I'd take a blank piece of paper and see if you're able to do that. So let me move back up here. You can hit pause for a minute. Let me see if I can erase. No, it's not going to let me. Okay, so here is your objective statement. See if you can do exactly that. The source, substrate, product, and optimal pH for one amylase, protease, and one lipase. And the very last thing that we need to do is talk about exactly how those enzymes are working. Now the midterm, a lot of people struggled with this, so we'll revisit this idea. Oops, here we go. Of hydrolysis. Okay. So in a hydrolysis, or in any kind of condensation hydrolysis, we're either adding or taking away water to either make or break the bonds between different monomers. And I'm going to give you the example of a polypeptide. Actually, a dipeptide, because I'm only going to be using 2. So one amino acid has the general formula of a carbon in the middle, an H and H, a C, which is double bonded to an O, which is attached to an OH, an R group, that's what makes them all different, and a hydrogen. And you can draw this either way. Typically you'll probably see the N and the C on the opposite sides. I flipped them, and that's okay. So one amino acid here, I'm going to draw exactly the same thing. Okay, so now I have 2 different amino acids next to each other. And so what needs to happen is I'm going to take this right here, hydrogen, hydrogen, and an oxygen, and look at that. Now we've got an H2O. So that's going to disappear. Okay. And you're going to create a bond between the nitrogen and the carbon, meaning that our dipeptide looks like this. And drawing it a little bit more appropriately. I'm just kind of moving this around so that it fits better. Is now we've got a hydrogen up here and double bonded to an O. Our enzymes are going to come in and act on this peptide bond. Okay, so instead of taking out water, we're going to be adding water. So change the way this arrow goes. We're going to break this bond right here, add in the hydrogen and the OH from the water, right? So a hydrogen and then another oxygen plus hydrogen are going to reattach to the amino acids here to break up the dipeptide to create two different amino acids. Or, you know, if this chain were to continue on that way and to continue on this way, then we've taken a long polypeptide and broken it into shorter polypeptides. Okay? Exactly the same principle goes for basically all of our different macromolecules. You could start with your fatty acids and your glycerol combining to form lipids and breaking them down exactly the same way. And our very last objective is to talk about, now we've had this food, we've swallowed it, we've digested it, we talked about what the different enzymes are that are going to break down lipids and proteins and carbohydrates. And so we've broken it down, and now finally everything has been chemically digested, right? We're not done yet though. The most important part isn't just digesting it, because then all you're going to do is you're going to take this food, it's going to make it through the digestive system, and that whole process, all of that energy that it took to get your food, first of all, and then digest it is just going to be pooped out. Very, very pointless. So we want to take as many nutrients as we can, and so we need to talk about exactly how that is done. And that's the process of absorption, right? So first we digest, but now we need to absorb, right? Exclamation points on there. And that is mostly going to be happening in the latter part of the small intestine called the ileum, which we alluded to a little bit before. So I'm going to look at the ileum kind of from a diagrammatic viewpoint, and it's not the best diagram ever, but here we go. And so what can we see on this? We can see the outer layer of muscle, right? And this is going to help with peristalsis and moving things throughout. There's a second layer of muscle in there, but this is where it gets interesting. We've got these folds all along here, right? And these are called villi, and actually these folds have villi on them. Let me show you what I mean. If I kind of come down here a little bit. Okay, so we've got the outer layers of muscles. Nicely drawn. And then we've got folds. Okay, if we zoom in even more, so here's the lumen. This is where food is going through. So here's our food. These folds have folds on them. Each of these folds is called a villus, or pluralized is villi. Let me fix that. Okay, so pluralized we have villi, but a villus. Now, our villus has a couple of key characteristics, of course, increasing surface area, but there's a lot of things going on so that food can go from the digested form in the lumen and be absorbed. So let's zoom back out here and actually take a look at a micrograph of a villus. Okay, first thing you should notice is that there are a ton of blood vessels in here. So let me change my color here. Okay, so right here is one villus. Now coming up through the center of the villus is a structure called a lacteal that I'm drawing in green, and you can't really see it. It's part of the lymph system. It'll be involved in absorption of fats, Not the most important piece of information right here, but certainly one that you should have heard of. So this is your lacteal. But the really good part of this, the real juicy part, as it were, are all of these capillary networks, all of these blood vessels, and you've really increased your surface area to volume. You've really increased the total area where you can absorb nutrients. So we might have some nutrients out in here, and our job, let's see if I can click on them. I don't think I can, but our job is to get those nutrients into the blood system. So let's take an even closer look. If I slide over here, here's my lacteal. My lacteal is made up of just one cell thick, like this. So that means for a nutrient to be able to pass from the lumen into the bloodstream, it's only going to need to go through two cells. One, which are on the lacteal, okay, so a cell right here. The second cell is going to be the cell that goes into the actual capillary, okay. So we've got a capillary network here, and so this would be showing you the inside of the capillary. Let me zoom in some more. Okay, so here we have the inside of the capillary. This is where the blood cells are. This is where the nutrients need to get to. And this here is also one cell thick the whole way around. Well, the villi have cells that are specially adapted to this whole process of really absorbing as much as possible and actively transport it from the lumen and into the blood system via the capillary. Okay, so each one of these cells has extra folds on the membrane, like this. These are called microvilli. So you've got microvilli on each of the individual epithelial or skin cells. Epithelial just means skin. So you've got these microvilli making up the lumen surface of the cells, and then of course you've got your nucleus and then a ton of ribosomes, and then the real kicker here is you've also got a ton of mitochondria, because you are going to need a ton of energy to do the active transport required in taking amino acids or fatty acids or lactose, for example, and moving them from the lumen where they've been digested and getting them into the cells, pumping them through the cells, in through the capillary network finally, and into the blood system. So congratulations, we have now absorbed our nutrients. Okay, that's a lot of work, but we're not done. The very last piece is not just to absorb, but to assimilate. And very simply put, it means taking something that was not human and turning it into human. So we've got our amino acids. When we assimilate them, that just means we're turning this into a protein, which is distinctly human. It is a protein that is necessary for your body, and voila. That is the entire purpose. So all of this presentation of taking in food, of eating and chewing and grinding and churning and digesting it chemically, and all the different chemicals that we've been using, all to get them down to their simple component parts, get them into the blood system. The whole purpose of all of that wasn't just to get it into the blood or to get it to the cells, but actually to turn it into some usable protein or usable structure. So congratulations, you've made it through your first video, and hopefully I will have a chance to see you Monday, tomorrow, if it doesn't snow. Good night.